Good evening, myself Shubh of group 322 and my topic is deciphering of genetic code and its meaning. A direct way toward the solution of the problem was suggested by Nierenberg and Matthew in 1961 upon introduction of synthetic polynucleoside into a cell-free system. The amino acids are incorporated into the polypeptide chain. The cell-free system contained ribosome, a set of tRNA, ATP, all necessary enzymes, but all did not contain DNA and mRNA. The ribosomal and supernet fraction were isolated by the centrifugation. The ribosome were washed off. The supernated liquid containing enzymes and tRNA was subjected to dialysis against a special buffer. To mixture of the two purified fractions was added an ATP generation system. A source of the energy synthetic polynucleotide were injected into the cell-free system. The incorporation of carbon-14 labeled amino acid into the fraction insoluble trichloroacetic acid polypeptide studied. It turned out to be possible to deceive the biological system. The ribosomes interacted with synthetic polynucleotide instead of with a natural mRNA. It was found that the polyuninucleotide chain simulated the polycondensation of the phenylamine and polycarbon, perform the same function with respect to proline. The first codon were found. If the code is a triplet, then the codon U correspond to PHE and the codon CCC to PRO. Then there was a study the action to copopolymers to known as composition, but with a random sequence of nucleotides. The frequency of appearance of triplets of definite composition is unknown. For such compromission into the polypeptide chain of Phi, Lu, Ni, and also in the smaller amount of SN and Lys. As we can see in the table, the coding triplet obtained from the experiment with poly AU. However, such an experiment do not yet provide the complete deciphering of the code. It remains unknown which of the three codon AW, UAU or UUA does code for TYR, etc. In subsequent experiment, Nierenberg replaced polynucleotide with trinucleotide to known structure, so trinucleotide tRNA amino acid. The synthetic of polypeptide complex are formed in the system. The synthetic of polypeptide does not occur in the case since the trinucleotide imitates codon. The formation of complex allowed to read out for this alone. All tRNA that becomes consecutive bound the labeled amino acid must be studied. All 64 triplets were thus studied and amino acid of which they become bound were identified. The primary structure of protein is coded genetically and the three-dimensional structure of globules is coded in biological functional manner. The mutation replacement for the template we have seen. The hydrophobicity of the amino acid residue is very important to the structure of globules. Accordingly, mutation that strongly after the hydrophobicity of the residue must have a stronger effect on biological properties of the protein than mutation that alter the hydrophobicity. Allow slightly mutation of the first type are more determined to the existence of the species and the individual that the second. One might think that the evolution that has led to the existing code progress in the direction of increasing reliability of the code in the sense that the fraction of the more deterioration mutation diminishes. Let us see how reliable the code is. The average difference in this value upon the bilateral replacement of one amino acid by another is 5370 joule per mole within the conditional classification of amino acid into two classes, the hydrophobic, the first 10, and the hydrophilic, the second 10 amino acids. The average difference are 3370 and 1640 joules per mole respectively. The class of amino acid depends primarily on the second nucleotide Y in the XYZ codon. With Y equals to U, the residue is always hydrophobic. Let us call mutations that do not alter the class of amino acid residue correct. Those which do not alter the class incorrect. The latter are more deleterious to the structure of protein. Replacement of one of the nucleotide in codon XYZ gives rise to the following distribution of correct and incorrect mutation. Replacement of X are given in the diagram, or there are altogether 350 66.5% correct replacement and 156 33.5% incorrect replacement. The correct replacement includes silent mutation. For example, those cases where the replacement of nucleotide is not accompanied by the replacement of residue. Because of the degeneracy of the code is built so that the probability of mutation is twice as high as the probability of an incorrect mutation. Let us calculate the average difference in the hydrophobic of the residue that correspond with the single replacement of nucleotides. We obtain the following answer. Upon the replacement of X 4200, Y 5370 and Z 1420 joule per moles.